Thank you for your interest in cell engineering at Transposon. This presentation will cover improving recombinant protein and bioproduction in cells. Mammalian cellular systems are effective for the production of therapeutic recombinant proteins. The problem is these systems are not perfect. Stable integration and expression at high levels is inefficient, and incorrect processing and/or modification of the desired protein product can be an issue. The solutions we'll discuss on next-generation genome engineering utilizing the piggyBac transposon and PB gold stable transfection kits, as well as engineering of the cellular systems to improve their capacity for protein production in stable and functional. A little background on the piggyBac transposon and how it works. First, you clone in your gene of interest. This could be a therapeutic protein, an antibody, into the piggyBac transposon vector, which is flanked by inverted terminal repeats. Then you co-transfect with the piggyBac transposase, which can be a DNA plasmid or mRNA. The transposase recognizes inverted terminal repeats from the plasmid, cuts the gene of interest and the transposon out of the plasmid, and integrates it stably into the genome at a very high efficiency. So one of the main advantages of using the piggyBac system for stable cell line generation is that it's extremely efficient. Single transfections make stable cell lines with extreme efficiency. It works in all mammalian cell types, all mammalian genomes, and other species. A wide variety of cell lines have been demonstrated. These are just a few. And here this image shows the percent of CHO cells expressing GFP after 90 days. This is a pool of cells. So this is a single transfection, no selection, no cloning, no screening. And you can see that over 90% of all these cells in this pool are expressing GFP after 90 days. So using this system, you can create stable cell lines 10 times faster than competing technologies, and you can bypass those time-consuming and expensive cloning and screening steps. piggyBac has an unmatched stable integration and expression capacity. This image shows expression in TRAMP C2 cells over time using plasmid, lentivirus, or piggyBac. And you can see that over time the expression is completely lost when plasmid and lentivirus integration is used. However, over time, the piggyBac stable expression is rock solid. So when used for bioproduction, all of these advantages can be utilized. The piggyBac transposon can be utilized to integrate recombinant protein genes, and this shows that when using the piggyBac system, you get a much higher level of expression and production using in CHO cells than using traditional plasmid technologies. So more efficient for recombinant protein production and antibody production. So moving from stable expression and integration to targeted engineering of mammalian systems, we start with the most effective, flexible, and precise targeted nucleases, our XTN talons and our next generation CRISPRs. We can design these to target any gene for either to go down the pathway of non-homologous end joining for frame shift deletions and knockouts, or we can also provide a donor vector with arms of homology for targeted integration and knock-in. One example of this was our targeting of the FUT8 gene in CHO cells, which we did in collaboration with Aerogen Biosciences. The FUT8 gene is responsible for post-translational modification. We targeted that gene with our nuclease systems. None of the fucose was shown on the cell surface, which indicated that the knockout did occur. And more importantly, the FUT8 knockout CHO cells demonstrated the expected 6 to 10x shift of increased ADCC activity, which indicated that the antibodies were being produced with the correct post-translational modifications. So the take-home message is that new genome engineering technologies can improve bioproduction systems. Transposon has custom reagents and technology portfolios. We can produce the reagents so that you can use them in-house. We can license technologies to you, and we also have custom genome engineering services with multiple advantages. And you can also leverage Transposogen's intellectual property portfolio for freedom to operate in commercial projects. Thank you.